Hello everyone, my name is Mahjabin and my ID is 2621567. Today I will talk about DNA sequencing. I will explain that DNA sequencing is how it has changed over time and what its future look like. First, what is DNA sequencing? DNA sequencing is the process of finding the exact order of the four DNA bases A, T, G, and C. These stand for adenine, thymine, guanine, and cytosine. You can think of DNA as the instruction book of our body. DNA sequencing helps scientists read this instruction book. It is used for digest, diagnosis, medical research, and many biological studies. Today, modern machines can read DNA quickly and save the results as digital data. Before learning the technologies, let's understand three important terms. Oligonucleotide is a short piece of DNA. It helps start the sequencing process. Plasmids and vectors are small DNA carriers. They carry DNA into bacteria like Escherichia coli, where many copies of the DNA are made. Back vector or bacterial artificial chromosome can carry very large piece of DNA. It was very important in the Human Genome Project. DNA sequencing has developed through three generations. The first generation is the Sanger method created by Frederick Sanger in 1977. It is very accurate with more than 99.9% accuracy. But it is slow, expensive, and can only read short DNA pieces. Even so, it was the main method used the Human Genome Project. Sanger sequencing. Sanger sequencing has three main steps. Step 1, DNA preparation. The target DNA is placed into vectors and copied inside bacteria. Step 2, sequencing reaction. The DNA strands are separated by huge primers attached to the DNA, and DNA polymerase builds new strands. Special fluorescent DNTPs top the DNA chain at different pages. Step 3, capillary electrophoresis. An electric field separates the DNA fragments by size. A laser reads the fluorescent colors and finds the DNA sequence. Second generation NGS. The second generation is called next generation or NGS. This technology was big improvement. Instead of reading one DNA pieces at a time, NGS reads millions of DNA fragments at the same time. It uses breeze amplification and fluorescent detection. Because of this, DNA sequencing became much faster and much cheaper. Third generation sequencing. The newest technology is third generation sequencing, also called long read sequencing. Unlike NGS, it does not need to cut DNA into many small pieces. It can read one long DNA molecule directly and in real time. Technologies like nanopore and PacBio make this possible. Some devices like the Minion are so small that they can connect directly to a laptop. Future of DNA sequencing. The future of DNA sequencing is very exciting. Scientists are working on single cell genomics to study one cell at a time, spatial transcriptomics to see where genes are active inside tissues, AI powered base calling for faster and more accurate results. Point of care diagnostics for quick genetic testing near patients. To conclude, DNA sequencing has changed from a slow and difficult process to a fast, accurate, and real time technology. It is helping doctors, researchers, and scientists improve healthcare and understand diseases better. Thank you very much for listening.